All right, let's dive into something that really blurs the line between biology, robotics, and what used to be pure science fiction. This is the story of how scientists are teaching machines to think by giving them a tiny piece of a human brain.
Now, when you picture a robot's brain, you're probably thinking of silicon chips, right? Complex code, wires, the usual. But what if its mind wasn't programmed, but grown? What you're looking at here, well, it represents that exact idea. A totally new kind of intelligence, one that isn't built from ones and zeros.
Yeah, you heard that right. It might sound like a movie plot, but in labs right now, tiny clusters of human brain cells are actually learning to control robots. This is the wild, groundbreaking reality of a brand new field called organoid intelligence.
So, how on earth is this even possible? Well, the whole journey begins with something scientists call a brain organoid. The easiest way to think of it is as a mini brain grown right there in a lab. The key phrase here is self-organized. See, scientists don't build these things neuron by neuron. They just create the right conditions, provide the right nutrients, and boom, the human brain cells assemble themselves into these complex 3D structures. They even start firing off signals and forming neural networks, a lot like a brain in its very earliest stages of development. It's pretty incredible.
The process itself is just remarkable. It all starts with ordinary adult cells, maybe from a simple skin sample. Those cells get reprogrammed back into stem cells, which basically gives them the superpower to become any type of cell. Then, with just the right biochemical nudges, they differentiate and self-assemble into a mini brain, often no bigger than a grain of rice.
Okay, so you have a mini brain in a dish. That's amazing all by itself. But now what? How do you connect it to the outside world? More importantly, how do you actually get it to learn anything?
This is where a device called a multi-electrode array, or MEA, comes in. It's the crucial link. The organoid sits on this grid of tiny electrodes, and it acts as a two-way street for communication. The array can listen by picking up the electrical signals from the neurons, and it can talk back by sending targeted electrical pulses to stimulate the cells. It's basically the ultimate brain-machine interface.
So this setup creates what's called a closed-loop learning system. Here's how it works. At first, the organoid's neurons are just firing randomly. A computer interprets one pattern of firing as, say, turn left, and tells a robot to move. If the robot does something good, like avoiding a wall, it gets a reward signal, a nice, stable, patterned electrical pulse. But if it messes up, it gets a punishment, a messy, disruptive jolt of electricity. And get this isn't just theory. Researchers at the University of Cambridge actually proved it works. They hooked up an organoid to a simple little robot, and over time, it got noticeably better at navigating a maze. Its neural activity, which started out as just random chaos, became structured and predictable. It was literally forming pathways dedicated to avoiding obstacles. It was learning.
This is all really amazing, but I get it. It might also sound a little unsettling. So what's the point? Why are we merging human neurons with machines? Well, the answer comes down to the absolutely unparalleled efficiency of biology.
Just look at the difference between our current AI and this new organoid intelligence. Silicon-based AI is powerful, for sure, but it's incredibly energy-hungry, and it needs enormous data sets to learn anything. The human brain, on the other hand, is the most efficient learning machine we know of. It learns from just a little bit of data and runs on only a few watts of power. Organoid intelligence is all about trying to tap into that incredible biological efficiency. How much more efficient are we talking? Well, startups in this space, like one called Final Spark, estimate that for certain jobs, these biological processors could be up to 1,000,000 times more energy efficient than our very best silicon chips. 1,000,000! In a world where our data centers are causing a real energy crunch, that's not just an improvement, that's a full-on revolution.
And this field is moving at an absolutely insane pace. I mean, the first brain organoids were only created just over a decade ago. The term organoid intelligence itself was just coined in 2023, and in the tiny amount of time since then, we've already got open-source systems and the development of even more complex whole-brain models. The acceleration is just breathtaking. But of course, this incredible power brings with it some profound questions. As these mini-brains get more and more complex, we are wading into completely uncharted ethical territory, and it's forcing us to confront some very, very difficult possibilities.
I mean, let's start with the biggest one. If an organoid can learn to avoid a negative stimulus, is it experiencing some primitive form of frustration or pain? We honestly don't know the answer, but we have a moral obligation to ask the question.
And that, of course, leads to the next logical question. At what point does a complex cluster of human neurons stop being just tissue and become something more? An entity with interests, maybe even rights.
Now, it's really important to ground ourselves in the current reality. Today's organoids are structurally very primitive. They're nowhere near achieving consciousness. But the scientific community is, very wisely, trying to get ahead of the problem. Bioethicists are already working with research teams, developing ethical guidelines to make sure this technology is pursued responsibly right from the start.
So, where does this strange and exciting journey take us next? Well, the path forward is really about making these systems more complex, more useful, and making sure we do it all in an ethically sound way.
In the near future, researchers are focused on some pretty clear goals. They're working on building more sophisticated organoids to create these amazing, dynamic models of diseases like Alzheimer's. That would let them study these conditions in ways they never could before. And a huge side benefit is that this could dramatically reduce our reliance on animal testing. There are still major technical hurdles, of course, but the momentum is just undeniable.
Ultimately, this isn't just about building better computers or smarter robots. It's a fundamental quest to understand ourselves. By teaching a piece of a brain to interact with the world, we might just learn what it truly means to be intelligent, to be aware, and maybe even to be alive. The journey has really just begun.